Hello, everyone, and welcome to today's webinar. My name is Susan Harrison-Uy, Director of Marketing at Vector Laboratories, and I will be moderating our discussion. Today, we are joined by Dr. Craig Powell, Director of Market Development at Vector Laboratories. Dr. Powell will be discussing ways to optimize your immunofluorescent staining to help you achieve consistent, reliable data. We like our webinars to be interactive. We encourage you to submit your questions and comments at any point during the webinar, and Dr. Powell will answer these questions during our Q&A session following his presentation. To ask a question, simply type your query into the questions pane and click submit. We will try to address as many of these questions as we can during the Q&A session. The webinar will be available on the Vector Laboratories website, and we will email you with a link to the on-demand webinar within a couple of days. Please note that you will not be able to download the presentation slides. We have also provided some useful resources related to today's webinar topic in the Handouts tab. You can access and download these documents at any time during the webinar. And with that, I'd like to introduce our speaker. Dr. Powell received a PhD in physiology from the University of Queensland, Brisbane, Australia, where he first learned histological techniques while studying reproductive biology in bats. Subsequently, he conducted his postdoctoral research at the California Primate Research Center at UC Davis, where he examined cellular migration and differentiation using tissue-based staining methods in early post-implantation primate embryos. Since joining Vector Laboratories 25 years ago, he has leveraged his deep technical expertise to help researchers optimize and troubleshoot their experiments. He currently serves as the Director of Market Development at Vector Laboratories. Dr. Powell?
Thank you very much, Susan. Good morning, good afternoon, and good evening to those who've had the opportunity to join us today from around the globe. As Susan mentioned, today's webinar is about optimizing immunofluorescence staining. Here's a brief overview of the topics we'll be discussing today. We'll look at sources of background interference, sometimes referred to as non-specific staining. Then we'll break that down a little bit. We'll look at autofluorescence specifically and ways and treatments that people have implemented to reduce and eliminate autofluorescence. And we'll have a look at some case studies that really underscore how prevalent autofluorescence is in these immunofluorescent-based assays. Then we'll look further afield into additional troubleshooting beyond autofluorescence. Then we'll finish up with trying to retain the signal that you have worked so hard to achieve. Okay, so let me just walk you through this particular image I've got here, because I will be referring back to this further on in the webinar. So this image here is a fairly standard one-step detection methodology for immunofluorescence, whereby this surface here could be a tissue section or a cell, for example. This particular white dome is the target antigen that is being bound here by an unconjugated primary antibody and then subsequently detected with a fluorophore conjugated secondary antibody. So just keep this particular schematic in mind as we walk through the webinar a bit. But this really showcases where background interference can come from in your immunofluorescent assay. It may come from the detection reagents, which include things such as the primary antibody and the secondary antibody, or indeed even blocking reagents, or it may be inherent in the specimen or endogenous autofluorescence, and we'll get into that momentarily, or indeed it could be a combination of both the detection reagents and the tissue components themselves. There's some of you who might be new to the area might be thinking, well, why would this be particularly problematic if it's not as abundant a problem in a given specimen? Well, I think you have to take a step back here and look at some of the potential problems that could arise. One, if you've got a weakly expressed antigen, and then you have a very highly expressed background autofluorescence, for example, or non-specific staining, that could obscure the target antigen localization and blanket that over. Also, background interference leads to interpretation of false positives. Perhaps you're working on a treated specimen and you're not completely clear on exactly where the treatment would be influencing the target antigen upregulation, for example. You could interpret some of the background staining as being positive staining, but of course then it would be a false positive and you'd be reporting that incorrectly. And lastly here, the assay validation. Background interference throws into question the credibility of the data that you're reporting on, and also creates a lot of problems with reproducibility, both at your end, if you try to reproduce it either in the same specimen or a different tissue block, or others who want to build on the results that you're reporting. Now, some of you may have experienced background before and been a little frustrated and just thrown everything out. Bought new reagents, cut new sections, and found that the problem still exists. So for something like determining background, you need to do effective troubleshooting. And really it's a two-step process, as I've indicated here. You have to identify the source, or indeed the sources of background, where it's coming from. And then once you've done that, you have to put into the workflow the specific methods to eliminate that source, or indeed sources of background, and give you a clear, unequivocal view of the target you're trying to identify. So let's have a quick look at what is autofluorescence. And I've got an image down the bottom here that showcases quite nicely something that you may have seen at your end. Very prominent red blood cell staining. Maybe there's interference from collagen or elastin, or indeed due to things such as formalin fixation or aldehyde-based fixative toning over the section. So autofluorescence is fluorescence inherent or endogenous to the tissue or cell, or indeed due to the use of certain fixatives. And this background fluorescence affects the signal to noise ratio. And what I mean by that is the noise is the background and the signal is the target signal that you're trying to report on. And really what you want in a best case scenario is no background or no noise and a very high signal to give you an unambiguous view of what you're trying to look at. Now, some investigators might be thinking, well, how do I even know that autofluorescence is present in my preparation? And that's quite easily done by examining the actual specimen you've got. And I've duplicated here this image just from the prior slide, whereby this is an unstained tissue section. What I mean by that is it's been simply cut on a microtome. It's been mounted onto a glass slide, and then you can just visualize that under a fluorescence microscope. And the question I've got here is, do you see any fluorescence? And if you do, well, then certainly that would be referred to as autofluorescence. Is the autofluorescence present in just a defined spectral wavelength? And what I mean by that is it just in the green area? Is it in the red? Is it in the far red? And that's a fair question to ask. If it's ubiquitous and all over the spectrum, you're going to have to address that because you can't avoid it. But it might be that a certain autofluorescence may only be present in a certain defined wavelength, such as the green. That might allow you to then move into the red or the far red area and avoid the autofluorescence in those other wavelengths. That I would say I would recommend that with a degree of caution, in that then you're assuming that that autofluorescence would not follow through or follow you to those other wavelengths once you start cutting further into the block or look at an adjacent specimen, for example. It's also important to look at the fluorescence you've got, that autofluorescence. Is it a general toning, such as that presented in the background of this given slide here? Is it very prominent, such as the red blood cells? Or indeed, does it have a different appearance? And What I mean by a different appearance is it could be from a different source of autofluorescence. What we've just looked at is autofluorescence from largely tissue elements such as the red blood cells, the collagens, etc., and due to aldehyde fixation. Another source of autofluorescence is due to something called lipofuscin, which I've showcased over here I got from this particular paper. These dots here, highlighted by the arrows, are in exactly the same location that investigators were trying to identify certain markers. I think it was a serotonin. We'll get into lipofuscin a little later on in the webinar, but at this point, we're going to have a bit of a deeper dive into looking at eliminating autofluorescence from these given elements. So I'm revisiting this particular image up here, this untreated section, and what we have are some adjacent serial sections with different treatments. So investigators have tried to address autofluorescence for many years, and these are what I'd refer to as home brews, for example, or more traditional treatments. The one that stands out is sodium borohydride down here. Sodium borohydride has been showcased in a number of publications, Journal of Neuroscience Methods, for example, I think Journal of Histochemistry and Cytochemistry. The thing with sodium borohydride, actually there's a couple of things with sodium borohydride. One, it's rather toxic to make up. It does bubble quite a lot, and it's got a very short half-life in terms of its use. So you have to make it up fresh quite frequently. And then, of course, as you can see, it's relatively ineffective in quenching autofluorescence, either from the tissue elements I mentioned, or fixation, or red blood cells. Another commonly used methodology is using copper sulfate. Now, apart from being a heavy metal solution, the copper sulfate is effective in reducing some autofluorescence when it is bound to the tissue. But when you do subsequent buffer washes, the copper is removed and the autofluorescence tends to bounce back up again and reduce that signal to noise ratio. So that's an inherent issue with the copper sulfate solutions. Another approach investigators have taken is to look at certain formats of ink-based materials. This one here, as you can see, not particularly effective in reducing the red blood cell autofluorescence, and you still see an overall toning of some of those other tissue elements. The ink-based reagents, though, are effective, or some of them are effective, in quenching lipofuscin rather than these cellular-based reagent, cellular components. And as I mentioned, we'll look at the lipofuscin a little later on in the webinar. So what's to be done then if some of these more traditional or home brew methodologies aren't effective in reducing autofluorescence? In this particular slide, it's a little busy, but it shows three things. One, it shows from the initial slide, I asked you to have a quick look at the problem of autofluorescence due to these endogenous elements or cellular components or tissue components. It introduces a treatment called TrueView that specifically reduces and eliminates the background fluorescence due to those components and gives you a look at the workflow of how this particular product or solution is applied. So TrueView is applied after the immunofluorescent detection methodology. So if you remember back to the original slide, this is your target antigen, your primary antibody detected by a fluorescently conjugated secondary antibody. And with the presence of the autofluorescence, you'd apply TrueView, which specifically targets that source of background interference, but retains the specific signal on that secondary antibody. TrueView is a solution made up of three components listed as A, B, and C. It is a blue colored solution as showcased on these given images here, but you drop it directly onto the tissue section. It does turn up blue, which does not interfere with any of the signal. A quick buffer wash, and then you move on to mounting the tissue. Hopefully that gives you a little bit of an idea about the treatment. It's about a 2 to 5 minute treatment on that tissue section. And for those of you who may not be familiar with its action, I'll run you through a couple of slides here. So these are sections of human kidney, which is what I'd call a particularly problematic tissue with regards to autofluorescence. This is a tissue section up here in the green channels. This is a cut tissue section, no staining on it, and it's simply been cover slipped with a mounting media containing DAPI so you can see the blue nuclei. So there's no specific staining here. All of the green you see is due to inherent autofluorescence due to those components and fixation. And this is a similar section, human kidney, showing red autofluorescence in that channel. And just to showcase the treatment of autofluorescence, the TrueView treatment and the reduction of autofluorescence in those corresponding serial sections, it shows elimination of the green and in the red channel, that autofluorescence, but you can still see the blue nuclei, which gives you a very nice opportunity to identify the underlying tissue morphology. Here are some more subtle examples of reducing autofluorescence. And this might be something that you're familiar with. You might think, okay, I can certainly see the green staining here of the insulin in this particular islet, and I can live with this background out here of autofluorescence. But indeed, I think this is an opportunity for you to see if you can further optimize that signal to noise ratio with something such as a TrueView treatment. And this enables you then to identify some of these outlying satellite cells that might not be particularly evident in this untreated section. And the idea of these slides is not just to show you some pretty pictures, but for you to maybe just have a think about the specimen you're working on, and can you actually further optimize the signal to noise ratio at your end and get a more defined, clearer result. Here's another example whereby we've got some human colon stained for cytokeratin, which is the green in here, and then counter stained with DAPI. But unfortunately, you have all of this outlying, looks like vasculature interference out here, which does take away from the image itself. With TrueView treatment, for example, it would eliminate that and give you a much clearer, more defined, specific staining. And one further example here, which again is a more subtle autofluorescence interference, is human pancreas stained for cytokeratin. You can certainly see the luminal surface out here of the cytokeratin, the nice blue nuclei, and you can see ductal structures in here. You might be able to live with this autofluorescence in here, this connective tissue area, but it does detract away from that defined cytokeratin. And what I'd highly recommend is to just have a look at your preparation and see if there may be opportunities to fine tune it and get a much cleaner, less equivocal result at your end. All right, hopefully that addressed for you sources of autofluorescence and those cell and tissue components and fixation-based autofluorescence. Let's have a quick look at lipofuscin. And lipofuscin does present as a different appearance to that from what we've just gone through. And I'm referring back to this reference image that I showed earlier on, whereby the lipofuscin is in each of these given channels, the green, the red, and this blue channel here, and it looks like specific staining for these given target antigens. So this is where lipofuscin would be reported as a false positive, possibly. But lipofuscin itself is an accumulation of lipids and other material in aged mammalian central nervous system tissue primarily, such as brain and spinal cord. And these sections here are from a monkey spinal cord material. And if you're working on neurodegenerative diseases, Alzheimer's, Parkinson's, animal models like that, on those given tissues, you may encounter lipofuscin interference. And so what do you do in those given scenarios? Because TrueView, for example, does not reduce autofluorescence due to lipofuscin. But what I did mention earlier on was some of the ink-based reagents are effective in reducing autofluorescence due to lipofuscin. And I will say that I did get this image directly from the Biotium website. Here's the untreated section in the green, the red, and the far red channel. This is an ink format, the same one I showed earlier on. Seems to be effective in the green channel, somewhat effective in the red, but not so much in the far red. It seems to actually induce more fluorescence in the far red. Biotium does have a product that seems to be effective across the spectra in reducing lipofuscin autofluorescence. So importantly, if you do have autofluorescence, and again, if you remember back to that one of those slides I showed, how do you know if you have autofluorescence? Simply look at an unstained section and then have a look at what it looks like. Where is that tissue from? Central nervous system tissue is a very discrete punctate staining. It's probably lipofuscin, or if it's red blood cell related, for example, that might be due to some of these other cellular components. So identify the source of that autofluorescence and then implement the appropriate treatment to reduce it and hopefully eliminate it. Okay, at this point, some investigators might be saying, thank you very much for going through that. That seemed to be more applicable for standard immunofluorescent-based applications. I'm working with fluorescence on a different platform or a different methodology or technique, and you might be wondering whether or not something like a TrueView treatment would be effective on one of these different platforms or applications. So what I've done, I've had a little bit of a dig through some of the literature to show where other investigators have encountered autofluorescence, and it seems to be essentially ubiquitous through a lot of different fluorescence-based assays, and that they've successfully implemented a reducing treatment. And so these two papers here do refer to using TrueView in fish assays or fluorescence in situ hybridization. I'm sure you can appreciate from this image here that I've shown those defined areas on the chromosomes, obviously vitally important to identify. And if they had other dots due to autofluorescence around the place or interference, that would throw into question the assay data that they're generating. This is another example. These investigators were using a fairly established technique, tyramide signal amplification, and in combination with the Opal system from PerkinElmer. Importantly, these investigators were looking at infiltration of certain cells and antigens. I think it was PD1 in these given tissues. And you can see these arrows I'm trying to highlight here. You can imagine if there were other cells with interfering autofluorescence or background, that would diminish the results and cause confusion. So this is where TrueView was used in this particular application and found to be effective. RNAscope is a platform that is now under Bio-Techne from Advanced Cellular Diagnostics, and they did find background with that particular platform as well, and they used TrueView here on these particular organoids. Importantly, I wanted to draw out the fact that they did find that diluting the TrueView 1 to 10 in this given platform actually worked a lot better for them. I think they found that the strength of the TrueView, as per the instructions provided, quenched a little bit of their signal. So through the dilution, they were able to optimize it and get a great result at their end. One further platform or methodology that is gaining notoriety out there when investigators are looking at, say, spatial relationships, proximity ligation assays. And certainly if you're looking at this fluorescently, you may encounter background with that. Indeed, I was talking with some colleagues yesterday who are developing some PLA assays and are struggling with the autofluorescence issue that we've been describing. So I did find a publication whereby some investigators did use TrueView in this given platform, and it gave them a nice, clear result. So again, this is really just to showcase to you that autofluorescence is quite prominent and widespread in a lot of these applications, and that some of these treatments can be employed as well. I would highly recommend going to these publications or similar papers and look at some of the methodologies and how they've employed some of these treatments. Now, some of you might be wondering, I'm working on central nervous system tissue that has autofluorescence, but I also have autofluorescence from other cell-based components, or indeed red blood cells are quite prominent. Maybe it's an unperfused material. Therefore, I'm really trying to address any potential questions that may come up about using TrueView in combination with ink-based reagents. So I did have a bit of a dig through the literature, and I found out that indeed investigators are using TrueView in combination with some of these ink-based reagents. And I will say that the second paper I've got here probably spells out the methodology a little more clearly than the first one, but the image I took was from this first paper. But if you were looking for order of application and things like that, this particular paper was a little more detailed in the application. They put down actually True Black from Biotium first, did their assay, and then followed through with TrueView in their workflow before cover slipping. All right, hopefully that gives you some insight into autofluorescence treatments and ways to overcome it, and that it's just not something that you might encounter in standard immunofluorescence, but in a lot of different platforms. But investigators are using some of these treatments to successfully reduce or eliminate background. All right, if you've done a quick test already for autofluorescence and you find you don't have any, but you still have background, what do you do? And if you remember back to my original slide, it's probably due to the detection reagents. And I'm revisiting this first image, or one of the first images I showed on the webinar here, of this very straightforward one-step detection methodology. And if you find that background is coming from a source other than autofluorescence, the idea is you run a defined negative control. And if you've got a very simple, straightforward methodology such as this one step, you would omit the primary antibody, but put on the secondary detection reagent at the given concentration and see if you do get any background signal. For those of you who might be running a more involved immunofluorescent detection methodology, what I've shown here is something a little more involved, is a two-step whereby you have a biotinylated secondary antibody, which is then subsequently detected with a streptavidin and fluorophore. You would run that same first defined negative control, whereby you'd omit the primary antibody, but you'd put on the biotinylated secondary antibody and the streptavidin conjugate and see if you get any background. If you do, then you can address that. But I would also highly recommend running a further negative control. Omit the primary antibody, omit the biotinylated secondary antibody, but then just apply that streptavidin fluorophore component and see if that component alone is generating background. Now, you might be running some more involved applications, such as ones we looked at earlier on, the tyramide system, for example, or something like that. You will have to run further defined negative controls for each of those steps or amplification steps, detection steps that you're putting on in your given assay to help pinpoint the source of that background from those detection reagents. Obviously, I can't do justice to all of those particular methodologies at this point, so I am really just focusing on these at the moment. Now, if you do see staining from those defined negative controls I mentioned, if you're just omitting the primary antibody and you do see staining, it's more likely due to, say, species cross-reactivity of the secondary detection reagent, possibly inadequate or inappropriate blocking, or inadequate buffer washes, and then less likely due to these particular areas over here. So if you're limited on time, tissue, resources, focus on these particular areas first. On the second step or two-step methodology I showed before, if you do see staining after you omit the primary and the biotinylated secondary antibodies, but just applying that streptavidin fluorophore reagent, the staining is more likely than due to something such as detectable levels of endogenous biotin. So we'll go through some of the troubleshooting methodologies over the next series of slides. So if you do see staining due to a secondary detection reagents, I did say it was due to potentially species cross-reactivity, and you might be encountering things such as a species-on-species scenario, whereby you are applying, say, a mouse primary antibody onto mouse tissue sections and detecting that primary antibody with an anti-mouse secondary antibody. That secondary antibody may be picking up endogenous or inherent mouse IgG in that tissue preparation in addition to the primary antibody. If that's the case, you can look at commercial blocking reagents that are out there, or indeed one thing that investigators have done is try to change the species of the primary antibody to avoid that cross-reactivity. So instead of applying a mouse primary onto mouse tissue, you would use, say, a rabbit primary antibody or a goat primary antibody, and then detect with an anti-rabbit or an anti-goat on that mouse tissue to avoid that cross-reactivity. One other step that's a little more involved and has its own potential issues is to conjugate that primary antibody and avoid having to use, say, an anti-mouse secondary antibody on mouse tissue. Conjugating the primary antibody does require a little bit of biochemistry there to do it properly and clean up, and it may interfere with some of the binding of the primary antibody if not done appropriately, and would certainly reduce the sensitivity of the detection methodology. So just bear that in mind if that was something you were looking at doing. Closely related species, so if you do see background when you're applying, say, a rat primary antibody onto mouse tissue and detecting with an anti-rat secondary antibody, you may see some degree of cross-reactivity. I would recommend looking for a pre-absorbed secondary antibody. So if you are applying, say, a rat primary antibody onto mouse tissue, you would use an anti-rat mouse-adsorbed secondary antibody, whereby that secondary antibody has been run down a column previously of mouse IgG, and anything that recognizes mouse has been physically removed from that preparation. So it's just the anti-rat that's there with negligible cross-reactivity with mouse IgG. Another option would be in cases where that secondary antibody has a lower extent of cross-reactivity. Let's just say a hamster. You're using a hamster primary antibody on mouse tissue, detecting with an anti-hamster secondary antibody that might have, say, 6% cross-reactivity with mouse IgG. You can dilute that anti-hamster secondary antibody in buffer containing, say, 2% mouse serum. Let that sit for about 30 to 60 minutes on the benchtop, absorb up that stickiness, and then apply it to that primary antibody. And that should significantly reduce background staining due to that cross-reactivity component. Okay, inadequate blocking. We did look at that briefly earlier on there. We mentioned it. So just verify that you do have the appropriate blocking reagent down there. Sometimes investigators do juggle a number of different serums at their end. So if you do apply goat serum and you're applying a goat primary antibody and you put down an anti-goat secondary antibody, it might be that secondary antibody will be binding to goat IgG that you put in place there as a blocking reagent that itself might be bound to charged particles, macromolecules, Fc receptors, whatever it might be. So just make sure that you are using the appropriate blocking serum or reagent. Indeed, there are alternatives to serum if you're juggling a lot of different sera in your lab or you're looking for alternatives to animal-based blocking reagents. Fish gelatin has been used out there in the publications, or indeed there are plant-based reagents. There's one we do offer called an animal-free blocking reagent that you could look at. Some of those, though, may also introduce themselves background. Make sure that they don't introduce particulate matter or artifact that could be interfering with it. If they do, certainly either spin them down or filter them, remove that particulate matter, and you discover that through one of your defined negative controls, or indeed use an alternative. Adding detergent into the wash buffers or diluents also is very effective in reducing hydrophobic interactions. About 0.1% to 5% of something such as a Tween 20 or a Triton X100 are very effective and do reduce background. Inadequate buffer washes I've got here, if you're working on things such as whole mount material, an excised piece of tissue, embryos such as maybe it's a xenopus, a zebrafish, something like that, or thick specimens, something that would be larger or thicker than, say, 20 microns. It's cut on a microtome or something like that. If you're working on these sort of preparations, anything more than a thin cut tissue section, increase the number of washes. So if you're just doing 2 times 2 minutes, extend it out to 3 to 5 washes, for example, and extend the length of time as well. 3 times 5 minutes, 3 times 10 minutes on some of these thicker preps to remove the unbound material. And for the last one I've listed here, detectable endogenous biotin that might be picked up by the use of a fluorescent streptavidin conjugate. Use a streptavidin biotin blocking step in the workflow, which is usually applied before the primary antibody. All right, hopefully that gives you some pointers, a few little tips and tricks there in reducing or eliminating background from sources other than autofluorescence. So now let's finish up a little bit here, looking at target signal retention. And this series of images here really showcases the importance of applying an anti-fade mounting media. There is a slight reduction when you do apply the media, but not that significant that you can't generate a good image. But if you don't apply a media, certainly the signal will literally fade before your eyes and won't allow you to then archive or rather image that material. There are a couple of little caveats I'd recommend to look at before you do jump in and simply slap on anti-fade mounting media. And I know investigators really apply this right at the end, and sometimes it's applied without any given due thought or applied haphazardly. Have a think about the specimen conditions you've got there. If you are working on, like I mentioned just on a couple of slides before, whole mounts or thicker preps, for example, or something that might be in a well or a chamber format that will require a volume of media, consider using a non-setting anti-fade media. You could use a setting or a curing media on thin cut tissue sections, something less than 10 microns that will hold the cover slip in place for you, and that way you can look at it under oil without having to seal around the outside of the cover slip. So mounting media do come in a number of different formats, as I've highlighted, setting or curing versus a non-setting as well. So just be careful that you are using the appropriate format for your specimen conditions or the particular way the specimen is presented. Also, fluorophore stability and compatibility. Not all anti-fade mounting media work or are effective across all the spectra. So indeed, if you're not getting a good signal retention with a certain anti-fade at a given spectra, look for an alternative mounting media that either others have reported on getting good results, or indeed you can trial at your end. Also, long-term storage or archiving of specimens is something investigators want to do so they can come back, re-image an area that might be of interest, for example. And if you were going to keep the slides for any length of time, and the example I've got here is 2 weeks actually just at room temperature, sometimes you do see a reduction of signal to noise ratio or artifact starting to come in, such as bubbles or retraction. Just have a look at some other mounting media out there that might not introduce that artifact over that given period that you're wanting to archive that material for and enable clear imaging of your particular specimen. Okay, at this particular point in time, I'm going to wrap it up and say thank you very much for your attention, and I'll open the floor to any questions you may have.
Thank you very much, Dr. Powell. To our attendees, please continue to submit your questions via the questions pane. We'll jump right in here. There's a question on whether TrueView interferes with DAPI.
No, it doesn't. We actually offer TrueView with a mounting media with DAPI in it. Well, one thing, let me just back up a little bit in that some DAPI is a counter stain for those out there who are not familiar with DAPI. I did show quite a number of examples there of DAPI showcasing nuclei. Once it does bind to the nuclear chromat and it fluoresces blue around 450, 460 nanometers. If you're using DAPI at a very light or low concentration, there may be some reduction in signal if you do use TrueView. Okay, so I'm now going back on initially my no, but my point being that you can then modify the concentration of DAPI if you do see that, make it a little more concentrated so you don't lose any DAPI, and then apply the TrueView. Certainly, though, we do have that optimized in a kit, the catalog number SP8500, TrueView with a mounting media that contains DAPI at an optimized concentration, so you wouldn't see any loss of signal. So if you potentially are seeing loss of signal or reduction of your DAPI fluorescence, either modify that concentration of DAPI or look to the product I just mentioned.
Thank you. There's a question as to whether TrueView works with liver tissue. This person is working with both mouse and human tissue.
If the autofluorescence is due to the particular components we were describing before, liver, of course, is highly vascularized, and you're seeing autofluorescence due to red blood cells or aldehyde-based fixation, or some of the basement membrane areas around hepatocytes or something like that, or around the basement membrane of these arterioles or the collagens, etc., in the arteries, then certainly TruView will be highly effective in liver tissue in reducing background. Yes, both in human and mouse tissue.
Thank you. So you discussed TruView and using that to decrease autofluorescence here. Are there any other methodologies that you can use to reduce autofluorescence, for example, with sample preparation?
Yeah, so the autofluorescence is generally only discovered, I guess, once you've actually done the fixation, the prep, and you've cut the tissue section, and then you encounter the problem. Can something be done to a tissue before or during prep to reduce the extent of autofluorescence? Certainly, if you were, or should I say, had flexibility in the choice of fixative, you could potentially look at ethanol or non-aldehyde base fixatives such as acetone, which tends to be more of a precipitating fixative than a cross-linking fixative, and that might not induce autofluorescence if that is indeed the source of the problem. In your sample prep, and then once you've done the fixation in the acetone, you cut the sections, and that might have a reduced or eliminated autofluorescence. But for something like Lipofuscin, though, that's generally already in the sample before you even fix it or cut it, and that generally has to be addressed after the tissue is already on the slide. So generally, apart from the choice of fixative, there's not a lot you can do to avoid autofluorescence ahead of time.
How can TruView work with genetically expressed fluorescence? So I imagine this is GFP and mCherry and tags like that.
Yes, I will say that we have not done that ourselves in-house. That's resources we don't currently have. But I did have a look at some of the literature out there. Investigators have looked at preparations that are expressing GFP. They've detected it with an anti-GFP antibody and then used TruView afterwards. So I will say that in that given application, TruView was used, and they did not have any issues. For something like mCherry, yes, there are certainly some published references out there that show the combination of retention of the signal and reduction of background, with retention of mCherry signal and reduction of background with TruView use. So yes, one thing I could not completely establish was if you just have GFP itself and you're just trying to identify that signal and not detecting it with an anti-GFP, would TruView reduce that signal itself and cause issue in identifying it? I don't think it would, given the action of TruView against a lot of different fluorophores and the fact it doesn't reduce autofluorescence of similar traces. So while I didn't find that in the literature in my cursory search, I don't think that TruView would be a problem in using it just on GFP expressed tissue itself.
Does the hydrophobic barrier from PAP pens interfere with TruView? Do you need to remove this barrier before you treat or cover slip?
So to retain the reagent around a tissue section, investigators do put residue or pen residue from a PAP pen or an image pen or something, usually a wax-based material, to keep that reagent in place, reduce the volume required and things like that. These pen residues do have a certain degree of inherent autofluorescence in them, and they can be present or viewed under the fluorescent microscope at the same wavelength as your given specimen. I will say, though, that that image is not over the top of the actual specimen you're looking at. You know, if you're looking at it 5x under an objective or 10x, you can probably see it. But once you start to come in underneath, say, a 40x objective, you know, none of that external fluorescence is going to interfere when viewing the fluorescence on the specimen.
We have some people that are talking about asking questions about specific tissue types. So one person who's having a lot of autofluorescence in muscle, and another individual who is having problems with skeletal tissue. Are there publications, or do you have experience with reducing autofluorescence with TruView in these tissue types?
Certainly, I did read in one paper there were investigators working on cardiomyocytes, I believe, isolated material. It might have been an organelle by memory, and also some cardiac tissue, cardiac muscle tissue, whereby TruView was used successfully. What we do mention in the instructions is a 2 to 5 minute incubation time, and for the vast majority of applications, TruView is effective within that time period. There may be tissue types that are a little abnormal, or treated specimens whereby a longer incubation time would be beneficial in reducing background from those elements. So I would say that there isn't a given tissue that whereby TruView would not be effective unless, of course, its autofluorescence is due to lipofuscin. So it may just require a little bit of optimization with the incubation time of the TruView. And indeed, in one of those case studies I showed, if you're using RNAscope, indeed, even diluting the TruView has been shown to give an optimal result. But if you are struggling with high fluorescence in a given specimen, apply the TruView initially as per the instructions, and then leave it on longer and see if that helps.
Do you wash TruView after application?
Yes. The recommendation is to use a phosphate-based buffer, a PBS, fairly standard, 10 millimolar sodium phosphate, just for a couple of minutes on a thin cut section, and then you would mount with your mounting media.
Does the incubation time for TruView affect the signal other than reducing the autofluorescence?
It can. So that might be something you might have to work around a little bit on some platforms. And the one I just mentioned earlier was the RNAscope. It might be that with certain detection methodologies, TruView may also lower the signal intensity of the specific signal, as well as reduce the background fluorescence. So you can certainly modify the dilution of the TruView. You can try that. You could also reduce the time or incubation time it's been on there, or indeed sometimes you can modify the exposure time with the camera. Instead of, say, a 100 millisecond exposure, you could use a 400 millisecond exposure, and to bring that signal up without appreciably bringing up that background. So there are a couple of things or little tips and tricks there you can use in trying to optimize the signal with TruView.
We have a question about mounting medium for thick sections. Somebody's working with 30 micron tissue sections. Do you have a recommendation for a mounting media that might work with those?
Depends a little bit on how that's presented, if it's on a glass slide or if that is a floating material. But I take it this particular thick section has gone through an immunofluorescent detection methodology. And for something like that, you might want to use something like a gasket or a chamber slide. rather than trying to squish down a cover slip on top of that, and use a non-setting mounting media in something like that. That would allow you to archive it over time. You can certainly pop that into the fridge or something like that, and image that successfully. So I wouldn't go toward a setting mounting or curing mounting media with one of those thick preps, but as I mentioned, something like a gasket or a chamber slide with non-setting media would probably be effective.
In one of the case studies, you mentioned that they diluted the TruView 1 in 10. What, if you needed to do that, what would you dilute the TruView in?
Very good question. Very good question. Just a straightforward buffer. PBS would be fine.
And we just have time for one more question. Somebody asked whether TruView was effective in reducing autofluorescence from endogenous molecules such as NADH and NADPH.
Yeah, some of these, if I remember correctly, are enzymes associated with mitochondria or metabolically active tissues. Maybe going back to the liver there. And while I don't have first-hand data where we've run a known highly expressed NADPH tissue and shown TruView to reduce it, I will say that for something like that, it tends to express in the DAPI spectra, in the blue spectra of the 450 to 460 nanometers. In the absence of DAPI staining, we just see black on all of these tissues. We don't see any residual blue fluorescence due to those components. So I will say that through those given studies, yes, TruView is effective in reducing background due to some of those other components you mentioned.
Thank you very much, Dr. Pao. Unfortunately, that is all the time we have for questions today. There are a number of questions that we were unable to answer, and so we will follow up on those via email. If you have any further questions, please reach out to our technical support team at technical@vectorlabs.com. As a reminder, a recording of the webinar will be available on the Vector Laboratories website, and you will receive an email notifying you when it is available. On behalf of Dr. Pao and Vector Laboratories, I would like to thank you all for your participation in today's webinar and for sharing your questions and comments. Thank you, everyone, and goodbye.
Thank you.